Welcome everyone to Contact's third produce safety webinar in the series. As a reminder, this series will tackle a different fresh produce safety topic every month through a dynamic webinar series that will bring together industry, academics, and regulatory to solve some of produce's industry's biggest and most timely challenges. This series is part of a larger industry outreach and risk mitigation project called Contact, or scientific challenges and cost-effective management of risks associated with implementation of produce safety regulations. This project is supported by a specialty crop research initiative from the USDA National Institute of Food and Agriculture. Any opinions, findings, and conclusions or recommendations expressed in this presentation are those of the speakers, the panelists, and do not necessarily reflect the view of the U.S. Department of Agriculture. This webinar series, coupled with other research and extension efforts, is part of a collaborative effort across 10 different organizations to enhance produce safety knowledge and compliance across the industry. We are so grateful to our collaborators for their efforts and support in this webinar series. The webinar series will continue next month. Doctors Callie Neal and Michelle Daniluk are going to be tackling Cyclospora and fresh produce. They will address what Cyclospora is, methods of detection, and probably most importantly, they will talk and discuss about actions you can take today to minimize the risk of product contamination. This multi-year webinar series continues through May before we go on summer break. We'll address agricultural teas. We'll have our first regulatory guest with Ag Teas. This is really driven by what you all want. So let us know if there's things that you want to learn about. At the end of the webinar, you'll have an opportunity to comment. While the webinar series lets us make contact with you once a month, that's really just not enough for us. So please make contact with us on our socials. Our social media, we have wonderful content that's educational on Twitter, Instagram, or I would challenge all of you on this call to volunteer and take a tackle at clarifying the confusing, which would be a short video clip tackling one particular topic. Let me first introduce our speaker today, Dr. Donald W. Schaffner. He's an extension specialist in food science and a distinguished professor at Rutgers University. His research interests include hand washing, cross-contamination, and quantitative microbial risk assessment. He has authored more than 195 peer-reviewed publications and educated thousands of food safety professionals around the world. He is a fellow of the Institute of Food Technologists, the American Academy of Microbiology, and International Association for Food Protection, IAFP. He served as an editor for the ASM Journal of Applied and Environmental Microbiology from 2005 to 2020. He began serving as a contributing editor for the journal Food Microbiology and as an associate editor for comprehensive reviews in food science and food safety in 2020. Dr. Schaffner was the president of IAFP in 2013 to 2014. And in his spare time, which is unreal that he has free time, he co-hosts the Food Safety Talk and the Risky or Not podcasts. Don, I will welcome you. The floor is yours.
Laura, I really appreciate that. Let me go ahead and get PowerPoint loaded here, and then I'm going to go in presentation mode. So the topic here today is risk-based thinking for ag food safety. And when Laura and Michelle and others asked me to speak, of course, I'm happy to speak on this topic. And then the information about the ag water rule dropped, and they said, hey, can you cover that too? And I said, yeah, I think I can make that fit within ag food safety. And so that's what I'm going to try to do for you today. So again, I'm going to talk a little bit about what I mean when I say microbial risk assessment. I'm going to talk about the difference between risk versus hazard. I'm going to talk about key differences between risk assessment and risk management. If this whole idea of risk is new to you, and I'll emphasize this later in the talk as well, understanding the difference between risk assessment and risk management, if you don't understand it, it can really change your world. And it certainly, when I realized that there was a difference, it really put a lot of things in perspective for me, not just about agricultural food safety, but about food safety in general. I'm going to give a couple of examples of what do we mean when we say something is high risk, and I'm going to hopefully give you some examples that'll stimulate your thinking. I'm going to give you a practical example about modeling listeria, which is based on some work that I did for Center for Produce Safety. And then I'm going to do a risk-based look at the new proposed ag water rules. Now, don't get your hopes up. I want to lower your expectations right away. All I'm really going to do is walk through the high-level overview that FDA has already provided. But what I'm going to do when I do that is hopefully interject some director's commentary or some armchair quarterback commentary. That's probably a better analogy. I'm not directing anything, but as to how I think that the new rules really are risk-based. I look forward to having a very stimulating discussion afterwards where you guys can talk more about how you feel about the ag water rules and our debate with me whether they're really risk-based or not. Okay, so let's get into some definitions. Hazard versus risk. A hazard is the thing that we're trying to control, right? It's a biological, chemical, or physical agent in or condition of a food with the potential to cause an adverse health effect. So it's salmonella, right? It's a rock, a splinter, a hazardous chemical. Whereas risk is a function of the probability of an adverse health effect and the severity of that effect consequential to hazard in food. And I think I'm using the codex definitions here, and they're a little bit wordy. If I was going to say them, I would say them with fewer words and maybe a little bit less precision. But let's talk a little bit more about what this means, okay? Like I said, a hazard, it's salmonella, it's listeria, it's a splinter, it's a rock, it's a peanut protein, it's a pesticide. And if we have hazard-based thinking, we think these things are bad and we need to stop them and we need to not have them in our food. And so hazard-based thinking is fine, but it's very black and white. Either the hazard is there or it's not. And if it's there, we're in trouble. Risk begins to get into some nuances, and those nuances include these terms of probability and severity. So let's say, for example, if 1 in every 1,000 servings contains one cell of organism XYZ, and one cell has a 1 in 300 chance of causing illness or causing diarrhea, a 1 in 20,000 chance of causing hospitalization, and a 1 in 500,000 chance of causing death, that's different than organism ABC, where this organism is much more prevalent. 1 in 10 servings contains a single cell. One cell has a 1 in a million chance of causing diarrhea. So it's a much less risky organism. A 100 million chance of causing hospitalization, and a 1 in a billion, if I'm counting my zeros correctly, chance of causing death. And so those two organisms are going to be very different in terms of how you manage them. Organism ABC is going to be much more common, but much less severe, whereas XYZ is going to be less common, but more severe when it is there. And you can actually make some comparisons between these two. Again, I've used this slide a bunch of times. I'm not sure it's necessarily helpful, but basically, and so I'm not going to walk you through it in detail, and I'm happy to make the slides available for anybody who wants them. This is just an Excel spreadsheet where I've taken those probabilities that I read off to you across the top and the severity off the top there, the first 2 lines. And then I just look at, well, what if we're talking about 1 million servings? What if we're talking about 100,000 servings? What if we're talking about 10 billion servings? And how does that relate to illnesses, hospitalizations, and death? And what you can see, again, because organism C is much more common, but much less severe, it doesn't really have an effect until we get up to a much higher level of serving, even though there's many more servings that are contaminated with that pathogen. And now, spoiler alert, these fictitious organisms are loosely modeled based on salmonella and listeria. And C is much more like listeria, whereas XYZ is much more like salmonella. Okay, let's move on to risk analysis. Risk analysis is commonly divided or thought of as being composed of 3 different areas: risk assessment, risk communication, and risk management. Now, risk assessment, at least the kind that we try to do in my lab, is quantitative. And so if it's quantitative, we're asking the question, how big is the risk? What is the probability of finding the organism in a serving? What is the probability of illness if that serving is ingested? What's the probability of hospitalization or death? But also, just as important, what factors control the risk, right? And so if we've got an organism in food and we've got a QMRE model, we can say, what if we change this? What if we change that? How does that impact the probability of the organism being there or the probability of illness or what have you? And that quantitative risk assessment process is a scientific process. It uses science. It uses scientific data. It may use expert opinion if we don't have all the data that we need. It uses math and statistics, and it combines all of that information using some sort of computer program or some sort of a method to be able to like literally turn the crank to make a little simulation of the world in the computer to answer the question, if we vary this factor, how does the risk change? I'm not going to talk too much about risk communication, but risk communication is talking about risk with affected individuals. In a way, this entire presentation is me communicating with you about risk, but I'm not really a risk communication expert. And there are people that are experts, and these people study how people perceive risk and what factors will influence how risk is perceived. Risk management. Most of you folks who are on the call who are in the industry, you are, whether you know it or not, risk managers. And so you're the ones that are trying to deal with or manage the risk. And for those of you who are with government agencies, you are also risk managers. I would say risk management is a political process, but that's political, lowercase p, not a political as in political parties. And so risk management deals with what do we actually do about the risk? And that is not a scientific process. And where I think people get concerned is, or where they get confused is, they want the scientists, the risk assessors, to what should we do? And the risk assessors will say back to the risk managers, what do you want to do? How low do you want to make that risk? There is no such thing as zero risk. But what's cost effective? What is effective in terms of what we can actually achieve? It may be you could drive risks much lower, but then a head of lettuce would cost $100 and nobody would really want that. And so it's all about how can we best use the resources we have to manage the risks that we have in a way that works for everybody. So companies can earn a fair profit, consumer health is protected to the greatest extent possible. And that's really where the sausage gets made in terms of the risk management. Okay, so let's talk about different ways of looking at risk. We can talk about risk per serving, or we can talk about risk per year. Let's consider, again, another fictitious example, 2 foods. Food A is popular, but it's low risk. Food B is less popular, but it's quite high risk. And so let's say, for example, food A has a risk of 1 in a million per serving. But guess what? 250 million people consume this food 365 days a year, okay? Food B has a risk of 1 in 100 per serving, but it's only consumed by a fraction of a percent of the people that consume the other food. And again, these are loosely fictionalized examples where food A is listeria in pasteurized milk, because guess what? Sometimes because of pasteurization failure, listeria does show up in pasteurized milk. And food B is listeria in smoked fish. Listeria is quite common in smoked fish, especially cold smoked fish. But guess what? Not a lot of us really eat cold smoked fish. And so that the risk is, from that perspective, low. But let's turn this around, or let's look at another way of imagining this. And if I were to ask you, which is riskier, A or B, it really depends upon which graph you're looking at. These data in this graph are based on all of the information that I just told you on the previous slide. They're just graphed in a different way. The graph on the left-hand side is the cases per year. And you can see, and again, these are loosely fictionalized, but they work for purposes of illustration. You're going to actually get a lot more illnesses from food A because so many people consume food A, even though the risk per serving is relatively low. So if you're looking at cases per year across a country or across a product line, or it's a very simple product line, just 2 products, the risk from A is much higher than the risk from B. On the other hand, if we, instead of looking at the cases per year, if we ask ourselves, what if we're the people eating those foods? What's the risk per serving? Guess what? If we look at risk per serving, food B is much riskier. Now, you can't see it, but this is actually an Excel graph, and there is a very small bar on the A side that is at a level of 1 in a million. It's so small you can't even see it. It's indistinguishable from the baseline on the graph, but trust me, it's there. I'll show you the Excel spreadsheet. So which is riskier? It really depends upon what it is you mean by riskier and what you're using to compare those risks. Okay, so I'm going to change gears now, and I'm going to talk about, again, some work that I did for CPS looking at predictive models for managing listeria risk. And here we were interested in which fresh produce items would require time temperature control for safety. What are the appropriate time temperature combinations for controlling that risk? And specifically, again, we'll come back to our old friend Listeria monocytogenes. Under what conditions will Listeria monocytogenes increase and by how much? This particular project, actually, we went on and did some really nice lab work and modeling work in collaboration with Laura's lab. And we've got a couple of really nice publications on this. But this was some work that was done prior to those lab experiments being executed. And we just simply used pre-existing mathematical models to look at the amount of time it would take for a 1 log increase to occur in Listeria monocytogenes. And then we looked at some worst case scenarios. So the modeling tool that we used was something called Combase. This is a freely available modeling tool, and all you have to do is you have to register for an account. It's something that I spend on any given week. Every month I probably use it at least once, and some weeks I use it multiple times a day. It's a really nice way to go in and to get a rough idea of how you think a given pathogen will behave. It's got the most extensive suite of growth models, but it does have some survival and inactivation models as well. So we used data from Combase. We plugged in some different times and temperatures and different intrinsic factors to create this graph. And what this graph shows is the relationship, again, according to the Combase model, that the time in hours for a 1 logarithm increase in Listeria monocytogenes, basically as a function of pH and temperature. And you've got, for the scientists in the audience, you've got temperature in degrees C across the bottom. For those of you who work in temperatures in the United States who are not in scientific researchers, that's temperatures in degrees Fahrenheit. The two scales are exactly equivalent, so you just use whichever one is more comfortable for you. And then we've got different pH values on the y-axis. And you can see we've got some pH values for different foods. Now, again, remember, these are not experiments done with these actual foods. These are model predictions as if the organism was being held. And most of these experiments are done in broth, right? And again, the work that Laura and I did, we showed basically that for different food products, whole food products that were intact, not damaged, the Combase models were conservative. In other words, the actual growth occurred in the foods occurred less than the growth predicted by the models. But again, this is useful. So what does this actually tell us? What this says is that pH has a pretty profound effect on the time for a 1 log increase. And so you can see there's contour lines here. So the 24 line, that's 1 day. So if a pH temperature combination falls on that line, that means that it takes 1 day for a 1 log increase. And so if I can just extrapolate here, let's say the food pH is 6.5, and then we'll drop down. That looks like about 9.5 degrees C, or if we go up, that looks like about 48 degrees Fahrenheit. So at 48 degrees Fahrenheit, pH 6.5, you get a 1 log growth in Listeria in about 24 hours. And if you drop the temperature, if you go from same pH, if you go from 48 down to 42, you see it's about a 2-day increase, 48 hours until you see that increase. And then of course the pH influenced the contour lines such that as you lower the pH, depending on where you are, if you're up around 6 or 7, it doesn't really matter. But as you start getting below 6, you start to, at the same temperature, you start to see longer and longer times for that log increase. So this is a different perspective on the same problem, okay? And so here we fixed the pH at a pH of 6, and we took some real world data on the shelf life of romaine, which has a pH of about 6, okay? And again, don't pretend that this is about romaine, but this might be useful for managing risk of romaine. And so what does this say? This says that for the assumptions that we've made, if we store the food at 38 degrees Fahrenheit, which is 3.3 degrees C, you can see that at 21 days, okay, we see about a 6 log increase. And so if the shelf life is 21 days at 38 degrees Fahrenheit, then the number that we're managing against is 6.1. And so what that says is, okay, if we have already agreed that 38 degrees Fahrenheit is an acceptable temperature and it gives us a shelf life of 21 days, guess what? If we store it at 42 degrees, that shelf life is going to be 13 days. And if we store it at 45 degrees Fahrenheit, the shelf life is going to be about 9 days. And if we store it at 55 degrees, now our shelf life is, in terms of relative risk, is somewhere around 3 days. And so Part of the reason why we developed this tool is that we wanted to be able to address questions from retailers who said, hey, look, received the food and it was at 44 degrees Fahrenheit. Do I have to throw it out? And the short answer is no, or the science-based answer is no, but it might impact the listeria risk. And a simple tool like this will help us be able to use that approach to manage risk. All right, now we're going to change gears and we're going to talk about the new ag water rule. And so this was dropped on us on December 6, 2021. And the requirements in this proposed rule replace the pre-harvest microbial quality criteria and the testing requirements. And then what they've replaced them with is system-based pre-harvest agricultural water assessments. Okay. Now, FDA did not use the word risk. I threw the word risk in there, but then I crossed it out. And so what I'm going to do in the remaining slides is walk through the language. It's not the language of the rule, but the high level language, and talk about what this might mean from somebody like me who looks at things from a risk-based perspective. Okay, ag water systems, the location and nature of the water source, including whether it's groundwater or surface water, is important. Guess what? We already know that there's probably a difference in risk if you're using groundwater or you're using surface water. Water distribution systems, whether it's open or closed. Guess what? Again, we have information, either information or educated opinion, that a water source that's open to the environment is probably more risky than a water distribution system that's closed to the environment. And then the degree to which the system is protected from sources of contamination, other people using the water system, animal impact, one of the big major components of the work that we're doing in Contact. We have an entire research objective. We're looking at animal impact. Guess what? We have another entire research objective in the Contact project looking at biological soil amendments. And then, of course, we're not looking at human waste, but of course, we know that certainly if human waste were to get into an ag water system, that's probably not a good thing, even though most human waste doesn't contain pathogens. Some of it does, and that's going to end up in the water, and that's going to potentially end up on our produce. And so all of this language it's risk-based language. Now, again, it doesn't get into specific details, but it is pointing us down a risk-based pathway. Again, and certainly there's some research needs here. And again, I'm really glad that we're doing the work that we're doing in the Contact project to be able to look at this. Okay, ag water practices. We know either from data or from reasonable hypothesis that it makes a difference whether you're using sprinkler or spray, drip, furrow, flood, or seepage irrigation. And then, of course, we also know that when pathogens are there and they are applied, the time interval between the last time you applied water and the time when you harvest the produce is going to make a difference in terms of risk. Another interesting aspect of what FDA said is the crop characteristics. How susceptible is the produce to pathogens adhering on the surface, and to what extent would pathogens internalize? And I know the topic of internalization is a hotly debated one. My understanding is it's something that we see that can happen in the laboratory, but whether that actually happens under real world conditions or not, or the probability, or it's about probability and severity. So how does it happen and how severe is it? And again, these are research questions that have been answered to some degree and are open questions to some degree as well. We know, or at least we believe, that weather events or rain events can certainly influence the probability of pathogens ending up in food. Air temperature, guess what? Air temperature is important. And as a little bit of a teaser, I'm going to show you a figure from a manuscript that I'm currently working on with Michelle Daniluk that's going to show the potential impacts of that. Again, we need more research like this to be able to make intelligent decisions, but at least we're getting other relevant factors. Again, remember, these proposals are replacing testing, but that doesn't mean that you still can't use testing. And again, as a quantitative microbiologist, I think it's always better if you have data, and certainly some testing results are better than no testing results. And of course, more testing results are better than fewer testing results. But again, the decision about how much testing to do, that's a risk management decision. Risk assessment will tell you the implications of that testing, but it's the risk manager that ultimately has to decide on what's my test budget. Do I have money for 100 samples or 10 samples or 1,000 samples? And how do I allocate those samples? That might be maybe informed by risk assessment. And then what do I do with those? That's going to be risk management. Okay, so again, what FDA has said is if your water is not safe, you need to discontinue use and take corrective measures. That makes perfect sense. That's a very sensible risk-based decision. If there's known or foreseeable hazards related to animal activity, biological soil amendments of animal origin, or human waste, then you need to implement risk mitigation measures promptly. And then again, no later than, as it says there, the same growing season. Again, more risk-based language here. If there are one or more known or reasonably foreseeable hazards not related to any of those things, and you should implement mitigation procedures as soon as practical and no later than the following year. Or you can test the water as part of your assessment, and then based on the results of the test, you can make the changes from there. And then again, if you determine there are no known or reasonably foreseeable hazards, then you just need to inspect and maintain the water systems regularly, ideally at least once a year. This is, for those of you who are familiar with HACCP, this is that step in the HACCP plan where if nothing changes, you're still going to review your system every year. So again, the language and the phrasing of this looks very familiar to people who've worked in these kind of risk-based systems. Now, whether everybody's happy with this or whether you'd rather default back to the testing that FDA was going to have before, that's an open question, and you're certainly welcome to your opinion. So I guess the question, as I begin to wrap things up here, what does the future look like? And maybe the future looks like this graph. And so again, this is based on some work that's as yet unpublished. We've got a manuscript in development. This is some work that Michelle's team did looking at spraying basically generic E. coli onto oranges. And then my team is handling the modeling part. And so what we did was we were able to take weather data from weather stations that were very close to the trees where Michelle's team applied the E. coli. And we said, okay, what does the weather look like? Is it hot and dry? Is it cool and dry? Is it cool and humid? Is it hot and humid? Okay. And then looked at what is the probability of generic E. coli surviving on those oranges depending upon the weather conditions. And so what you can see, that solid black line, that is the survival probability of E. coli. What this says is, guess what? If you're going to apply E. coli to oranges, maybe you should be a research lab, but if you're going to apply water of suspect quality, it's really not a good idea to do that when the weather in the foreseeable future looks hot and humid. Now, again, it's Florida, it's hot and humid a lot, right? So that may not be completely helpful, but certainly at least this will inform that decision-making. And so hot and humid is bad. Hot and humid is going to encourage E. coli to survive, but guess what? Cool and humid is better than hot and humid. And so if you have a choice and you can apply the water when it's going to be cool and humid, that's going to favor the reduction of E. coli. And then what's even better than cool and humid, if it's going to be cool and dry, then that's your next best bet. And then, of course, the best conditions for applying water of suspect quality, at least based on the data that we have from Michelle's lab in oranges in Florida in this experiment, is hot and dry. If it's going to be hot and dry, guess what? E. coli really doesn't like it, and it drops, the survival probability drops, I wouldn't say instantaneously, but very rapidly. So let me summarize here. And I think this is going to leave us with lots of time for questions, which I really enjoy and I look forward to. I hope that I've convinced you that risk-based thinking is more useful than hazard-based thinking. Risk-based thinking gives us some focus and it allows us to prioritize. If we use hazard-based thinking, then everything is a hazard, everything needs to be controlled, and we just don't really know what to do because we're overwhelmed by all of the things that we have to do. If we can use a risk-based system that looks at severity and probability, that's going to tell us, hey, like, here are some things that maybe have a high probability and high severity, but maybe they're really easy to implement controls for. So that's where we should spend our money. We should focus on things that are high probability, high severity. And once we've got those problems solved, move down the chain. Remember that risk assessment is never going to tell you what to do. It's only going to say, if you do this, we predict that the risk is this. If you do that, we predict that the risk is going to be this other thing, okay? Okay. But hopefully together, risk managers working with risk assessors, you can begin to make better decisions. And then again, the proposed ag water rules look to me to be risk-based. They might even be more risk-based than the old proposed rules. Because remember, some of the pushback, as I recall, around the old rules that included testing was, well, is that really relevant to my industry or my part of the country or my water supply? And in a sense, that was a more one-size-fits-all approach, whereas this approach, there's a lot more flexibility. But again, it's putting the onus on the operator to be able to do this assessment. And again, we need more data. If you ask a scientist if they need more data, their answer is going to be always that they need more data. If you ask a risk assessor if they need more data, they'd say, sure, I'd like to have more data. But in the meantime, if I don't have the data and you still need to make a decision, let's think about if we can substitute some similar data, or let's see if we can use expert opinion as a surrogate. And then let's try to do the best we can with the data we have to try to just move things forward. And with that, that brings me to my last slide.
Thank you so much, Dr. Don.
Thanks so much, Laura, for the invitation. It was an absolute pleasure.
Absolutely. Thank you. And we look forward to seeing you next month. February 9th is a Wednesday. So hope you can join us.